Hello and a very warm welcome to everyone joining today's Select Science webinar titled How to Pipette Correctly, an expert guide to techniques and best practices. My name's Thomas Casburn and I'll be moderating today's session. Pipetting is a cornerstone technique in the laboratory. Achieving consistent, accurate and precise pipetting relies on the right pipetting tools, technique, ergonomics, and most importantly, the skill and the expertise of the operator. In today's webinar, I'm delighted to be joined by liquid handling specialist Steve Beckett from Thermo Fisher Scientific, who'll be providing training and advice to help you advance your skills so you can handle anything. Throughout the presentation, there will be the option to answer some poll questions, so I'd like to encourage the audience to participate in these. In addition, following the presentation, we will be moving on to our question and answer session. So if you do have any questions, please feel free to submit these at any time during the webinar, and you can do this by clicking on the purple tab on the left of your screen and then typing in your question. Without further delay, I'd like to hand over to Steve to begin today's presentation.
Thank you for the introduction. Here is the plan of learning. We'll go through air displacement pipetting and the difference between air displacement pipetting and positive displacement pipetting. Choosing the right tip for the application. Forward, reverse and repetitive pipetting techniques and when to use them. Accuracy and precision in relation to laboratory pipettes. Factors affecting pipetting quality with air displacement pipetters. And in broad terms, cross-contamination prevention. So, how do air displacement pipettes work? Air displacement pipettes move liquids into and out of disposable pipette tips. They create a partial vacuum with a cushion of air between the piston and the liquid. Adjust the volume settings to move the piston to its final position. Press the operating button to the first stop. This expels a volume of air equal to the volume set. Immerse the tip in the liquid and slowly release the operating button. This creates a partial vacuum that draws the liquid into the tip. Press the operating button to the first stop again. This compresses the air above the liquid to force the liquid from the tip. Press the operating button to the second stop to fully empty the tip, often called the blowout. How do positive displacement pipettes work? The piston in a positive displacement pipette makes direct contact with the liquid or material, which eliminates any airspace between them and creates a constant aspirational force. Adjust the volume settings to move the piston to its final position. If you press the operating button fully, this moves the piston to the end of the pipette tip. Immerse the tip in the material and slowly release the operating button. This draws the material into the tip. If you press the operating button fully again, this moves the piston to force the sample from the tip. Now some quick tips. The pipette tip, and the liquid should all be at the same temperature, ideally between 20 degrees centigrade and 25 degrees centigrade for most brands of pipettes. Pipettes are gravimetrically calibrated in the factory using double deionized grade water. The selected tips should be certified free of contamination and be recommended by the pipette manufacturer. Pipette tips are designed for single use only. Once a pipette tip is wetted and used and dries out for any reason, It will never perform within spec again. Now, selecting the correct tip for the application. Standard pipette tips. These are tips recommended by the pipette manufacturer. These are used for multi-purpose use. They're not filtered, they're not sterile. And these are the tips which are usually used for the calibration of the pipette. Extended length tips. These eliminate tip cone contamination because it keeps the tip cone away from the sample. These are extremely in-demand tips at the moment because of COVID-related applications and keeping the pipette well away from the sample, preventing contamination. Gel loading tips. Precise sample loading under agarose. Standard tips again, but this time certified free of detectable endonucleases and RNA, especially for PCR work. Low retention tips. Reduces sample loss due to adhesion, particularly useful for pipetting master mix and reduces the wastage of master mix in the tip after pipetting. Solvent safe tips. These are carbon filters for aggressive reagents. Sterile filter tips for microbiology, genetics, and PCR applications. Wide orifice tips. These are purposely designed for cell culture applications, and also if you're pipetting difficult viscous liquids, you'll get less sample hang-up and more accurate pipetting. The distal end of the tip is designed to allow cells to pass through without causing shearing forces. For cell culture applications, only happy cells grow. And individually wrapped tips. These are sterile tips that are endonuclease free and are only used at the very time that they're needed, preventing contamination. Now, pipetting techniques. The forward pipetting technique. This is the most commonly used pipetting technique for standard pipetting. Use this method for aqueous solutions such as buffers, diluted acids or alkalis. It is commonly used when pipetting and mixing a sample or reagent into another liquid. Press the operating button to the first stop. Dip the tip to a maximum of 1 centimeter beneath the meniscus into the solution and allow the tip to fill with careful control of the operating button. Hold for a minimum of 3 seconds for aqueous liquids and a minimum of 5 seconds for viscous liquids. Withdraw the tip from the liquid. Gently press the operating button to the first stop to dispense the liquid. Wait 1 second and press the operating button to the second stop to completely empty the tip. Then touch off the tip and remove the tip from the vessel by sliding it up the vessel wall. The reverse pipetting technique for high viscosity or foaming solutions. Press the operating button to the second stop. Dip the tip to a maximum of 1 centimeter beneath the meniscus into the solution and allow the tip to fill with careful control of the operating button. Hold for a minimum of 3 seconds for aqueous liquids and a minimum of 5 seconds for viscous liquids. Withdraw the tip from the liquid. Gently press the operating button to the first stop to dispense the liquid and hold the button in position. Do not dispense liquid that remains in the tip. Remove the tip by sliding it along the vessel wall. Discard any remaining liquid along with the tip. Release the operating button to the ready position. The repetitive pipetting technique for pipetting the same volume repeatedly. Ideal for pipetting master mix into a qPCR plate, for example. Press the operating button to the second stop. Dip the tip to a maximum of 1 centimeter beneath the meniscus into the solution and allow the tip to fill with careful control of the operating button. Hold for a minimum of 3 seconds for aqueous liquids and a minimum of 5 seconds for viscous liquids. Withdraw the tip from the liquid. Gently press the operating button to the first stop to dispense the liquid and hold the button in position. Remove the tip by sliding it along the vessel wall. Repeat steps 2 and 3 to continue pipetting. We have recommendations for different liquids. For example, aqueous solutions, buffers, diluted salt solutions, etc. Use an air displacement pipette and the standard pipette tips, and use the forward pipetting technique. For more viscous solutions, protein and nucleic acid solutions, glycerol, between 20, 40, 60, 80, etc. Then we say there's a choice of pipettes you could use. You can use an air displacement pipette, using the standard or low retention or wide orifice tips, whichever gives the best result. And consider using the reverse pipetting technique. Pipette slowly to avoid bubble formation. Or you could consider using a positive displacement pipette with the standard tips. But of course, there are many other types of liquid. If you have a difficult application or have difficulty pipetting accurately under any particular application, then why not get in touch with us, and we will consider and get back to you in a short period of time regarding our recommendations. Accuracy and precision in regard to laboratory pipettes. We need to pipette 20 microliters into a series of tubes. And using a particular pipette, we find that we dispense 20 microliters on the first occasion, 22 microliters on the second, 20 microliters again on the third occasion, 18 microliters on the fourth occasion, and again, 20 microliters on the fifth pipetting. When we work out the average volume dispensed, we do find that on average, this pipette is accurate and dispenses 20 microliters. So it is capable of dispensing the target volume. So on average, the pipette is accurate. However, the spread of the dispensings is considered to be unacceptable. So we say this pipette is accurate on average, but it's not precise. The problem with this pipette is, well, there are several factors that could be involved here. The end user may not be using the recommended tips recommended by the pipette manufacturer. There could be excessive wear on the tip cone, or the pipette could be grossly contaminated and should be cleaned, serviced, and calibration checked. Now, with another pipette, we have a similar series of dispensings. Again, we need to dispense 20 microliters 5 times. And on all 5 occasions, the pipette delivers 18 microliters precisely. So when this pipette is set at 20 microliters, it delivers 18 microliters. So this pipette is not capable of delivering the target volume when set to the target volume. So it's not accurate. However, the pipette delivers the same volume time and time again. So it's a very precise pipette, but it's not accurate. The problem with this pipette is that it may have been subjected to shock at some stage, and the calibration characteristics have changed. This pipette should be calibration checked and probably calibration adjusted, making sure that the correct tips are used when the calibration is performed. And of course, when the pipettes are serviced and calibrated by experts, you would expect the pipette to be accurate and precise. And that's what we see with this graphic. It delivers 20 microliters precisely every time with very little variation. The pipette is both accurate and precise. Now, factors affecting accuracy of air displacement pipettes. Now here we have a pipette with the recommended tips, and we're holding the pipette perfectly vertical. And we are pipetting double deionized water. And the tip only goes to a maximum of 1 centimeter beneath the meniscus. And good pipetting technique is used. We do a series of weighings, and we find that the accuracy of this pipette is between 0.2 and 0.4%. And when we look at the specifications for that pipette, then that's well within accepted limits. So there's no problem with scenario A or scenario 1. With scenario 2, we're using exactly the same pipette. We're using the recommended tips. We're pipetting double deionized water, and the pipette is held perfectly vertical during the aspiration of the liquid. However, the tip goes down 3 centimeters below the meniscus. So that's 2 centimeters further than is recommended. A series of weighings is done, and we find the inaccuracy is between 0.6 and 0.8%. It's 300% more inaccurate than scenario 1. And the only difference is that the liquid has gone down too far into the liquid. Because when the pipette was originally calibrated, the pipette tip didn't go down more than 1 centimeter. But on scenario 2, the tip goes down 3 centimeters, and it picks up excessive liquid on the outside of the tip, which joins the liquid when the gravimetric analysis is done, giving a positive bias to the result. Now, with an inaccuracy of 0.6 and 0.8%, that pipette would fail calibration. And of course, if a pipette fails calibration, then it should not be used for routine work. And the only problem there is the technique. Scenario 3, the same pipette, the same recommended tips, and the same liquid, double deionized water. But on this occasion, the pipette is held at a 30 to 40 degree angle when aspirating the liquid. And also the tip goes down between 3 and 4 centimeters below the meniscus. When we do a series of weighings, we find the inaccuracy is between 1 and 1.2%, 500% worse than the original scenario. And the only difference is the pipetting technique. So it's absolutely vital for accurate pipetting that the pipette is held perfectly vertically when aspirating the liquid. And also the liquid should not go down more than 1 centimeter from the meniscus. Now, the effective temperature. Temperature differences cause thermal expansion and shrinking in the air space. After temperature equilibrium, the influencing factor is liquid density. Cold liquid is more dense and hot liquid less dense than room temperature liquids. And remember, pipettes are calibrated at room temperature on double deionized water. So when the end user is working at room temperature and pipetting double deionized water at room temperature, it should be no surprise to find that using good technique, that perfectly accurate results are achieved. And that's the blue line on the graphic. However, if the liquid is at 15 degrees centigrade, and again, this is double deionized water, let's just say that the water's been kept in the fridge and it's below room temperature, then the liquid is more dense than the liquid would be at room temperature. So using a pipette which is calibrated at room temperature with the same liquid, then less liquid is aspirated at 15 degrees centigrade. So 5 microliters difference will be seen compared with pipetting a liquid which is at room temperature. That's an inaccuracy of about 2% just from that one pipetting step. Conversely, if the liquid is hot, above room temperature, let's say 30 degrees centigrade, then that liquid is less dense than it should be. So when using a pipette which is calibrated at room temperature, you will pick up more liquid than you intend. In fact, 7 microliters difference at 300 microliters. So over 2% difference will be seen just because of the temperature of the liquid when it's aspirated with a pipette that's calibrated at room temperature. The effect of viscosity. In this experiment, 200 microliters of viscous liquid, glycerol, was pipetted 10 times by using both forward and the reverse pipetting technique. The pipette was adjusted for glycerol using the forward pipetting technique. Using the reverse method yielded a smaller deviation between doses with greater precision. Using the reverse method provided bigger doses because the liquid column in the tip was taller and pushed out a larger dose. Now, as you can see from the graphic, the blue line were the results obtained using the reverse pipetting technique. There is a much smaller spread of results achieved. However, there is a slight positive bias to the result. When using the forward pipetting technique, there is a much larger variation in the volume dispensed because you're trying to clear the total volume from the tip every time. Density. Density, or mass-volume ratio, affects the liquid volume that is aspirated into the tip, dead air volume, and earth gravity. The density of liquids also varies according to temperature. Now, on the right is a list of compounds which are often pipetted with an air displacement pipette, which has been calibrated with double deionized water at room temperature. But look at the densities of these liquids. 30% glycerol, e.g. PCR master mix, the density is 1.08 cubic deciliter. 100% glycerol, 1.260 kilogram per cubic decimeter. Whole blood, 1.051 kilogram per cubic decimeter. And ethanol, 0.789 kilogram per cubic decimeter. So ethanol is lighter than water, but the other compounds listed there are significantly more dense. So with these liquids, it would be well worth considering calibrating the pipette with these liquids before dispensing accurately. Another quick tip. Rinsing the tip 3 to 5 times with liquid to be pipetted improves accuracy, especially with volatile liquids. It helps to eliminate capillary action. If you're pipetting a liquid which is volatile, very often with an air displacement pipette, you will find that initially the liquid will drip from the bottom of the tip. If you rinse the tip several times before taking up the volume that needs to be dispensed accurately, the dripping will stop because it helps to eliminate the capillary action. Also, rinsing the tip provides identical contact surface for all aliquots. This is absolutely essential for accurate pipetting. If you calibrate a pipette, then you must pre-wet the tip or pre-rinse the tip with the double deionized water before you take up the first quantitative volume. If you use a dry tip, then you'll under-deliver and the calibration will fail. So pre-rinsing the tip provides an identical contact surface for all aliquots. It also reduces sample hang-up by pre-rinsing the tip. You get less liquid left in the tip after dispensing. And also, if you need to pipette a liquid that's been kept in the fridge, for example, is at 4 degrees, and you're using an air displacement pipette, which is calibrated at room temperature, then if you repeatedly rinse the tip with the liquid that needs to be pipetted, then it helps to equalize the air temperature inside the tip to that of the sample, reducing the error. Preventing cross-contamination in broad terms. If your pipette has been used, then you must consider the fact that it will be contaminated. You must always assume that the pipettes that are used in your lab are already contaminated to a lesser or greater extent. So therefore, a problem is preventing contamination from your pipette to your sample. And a good way of preventing this is to use filter tips, especially barrier tips. Barrier tips are filter tips which capture the aerosol and stop it passing through. We recommend that you regularly clean and decontaminate the pipettes. We recommend this is a daily process by swabbing down the outside of the pipette with 70% alcohol, with special attention being paid to the tip cone. And of course, if the pipettes are autoclavable, you should autoclave them regularly. Preventing contamination from the sample to the pipette in the first place, then again, filter tips are recommended. They work in both directions, especially using barrier tips, capturing that aerosol, stopping it passing through. We also recommend that you keep the pipettes vertical. Now, we know that when you're aspirating sample, to deliver it accurately, you do need to keep the pipette vertical. You should also keep your pipettes vertical when they're not in use by keeping them on a pipette stand. This helps to prevent contamination of the tip cone, and it keeps it out of the way and off the bench. You should also have steady control of the operating piston. Please make sure that you don't allow the piston to snap back when aspirating liquid, because that will virtually guarantee aerosol production and contamination of the inside of the pipette. And as mentioned before, always use a tip rack, preventing contamination, and also it will prevent the pipette from accidentally being dropped on the floor, which will affect the calibration, which may not be noticed until you look at the results and find them difficult to explain. Now, we offer pipetting seminars on pipetting ergonomics. You may be using the best techniques for a particular sample or application, but if you're not comfortable whilst you're pipetting, then it'll be difficult to get good results. So we do recommend the pipetting ergonomics presentation. And of course, we have the proper pipetting techniques presentation as well. You can find out more on thermofisher.com/pipetting techniques. Thank you for attending.
Thank you very much, Steve, for that excellent presentation. It's now time for our question and answer session. So if you do have any questions, please send them in now. And you can do that using the purple tab on the left of your screen. So we've had a few questions come in already. And the first one here for you, Steve, is, can strong acids and alkalis be accurately pipetted using air displacement pipettes?
Good afternoon, everyone. I hope you enjoyed the presentation. Yes, regarding pipetting strong acids and alkalis, they can be pipetted accurately using an air displacement pipetter, unless they're particularly viscous, in which case you would consider using the reverse pipetting technique. I would recommend that filter tips are used when pipetting strong acids and alkalis, because the fumes that may be emitted from those liquids will go into the tip cone and affect the inside of the pipette and dry out any lubrication and affect the O-rings inside the pipette. But in terms of using an air displacement pipetter, there should be no problem there if the liquids are aqueous.
Okay, fantastic. Thank you very much for that. Our next question here is, are filter pipette tips designed to accommodate reverse pipetting techniques?
Well, that depends on the brand. Some brands of filter tips are not designed for reverse pipetting techniques, whereas others are. So I would recommend that if you're going to be using reverse pipetting techniques, which we do strongly recommend for viscous liquids, then you do need to try samples of the brands of filter tips that you wish to use, because not all brands can cater for reverse pipetting.
Brilliant, thank you for that. Our next question here is, are electronic pipettes more accurate and precise than manual pipettes?
If the manual pipette is used with good technique and gained with experience, then an electronic pipette and a manual pipette will perform very, very similarly. An electronic pipette has got many advantages, though. It will have extra functions which a manual pipette doesn't have, which makes the task easier and less prone to fatigue while a person is pipetting. With an electronic pipette, the pipette performs the same way every time the pipette is used because it's controlled by the motor. With a manual pipette, all the different steps in the pipetting technique are governed by the technique used by the end user, and is driven by the piston and the thumb. So with an inexperienced end user, and it is possible to get better results with an electronic pipette, because a lot of the pipetting technique itself is governed by the pipette. Whereas with a manual pipette, every single step in the pipetting procedure is governed by the end user. So with an experienced technician, end user, they should be equally accurate. But some people will find electronic pipettes easier to use, and they also protect you from, to some extent, from pipetting fatigue, which will affect the quality of the results.
Absolutely, brilliant. Thank you very much for that. Our next question here is, how do you avoid the blowout from pushing air bubbles into your sample? I noticed this particularly when pipetting small volumes.
Okay, when using the forward pipetting technique, you take up the sample, you transfer the sample, and then you place the pipette tip just above the liquid in the tube or in the microplate. Try just dispensing to the first stop and touching off at that stage. That avoids the blowout part of the procedure. If you press down to the second stop, then you will be bringing in the blowout function of the pipette, and if the tip is below the meniscus, then you will be introducing an air bubble below the surface. So hold the tip just above the meniscus when dispensing, and dispense just to the first stop. That will avoid the formation of the air bubble.
Brilliant, thank you very much for that. Our next question here actually refers to the second poll question, which was to maximize accuracy. What position should you hold the tip when aspirating the sample? And the question is, why is slanting your pipette the most inaccurate? Is there ever a reason where slanting a pipette is the best option?
Well, for the vast majority of brands of pipette, the pipette is calibrated, holding the pipette perfectly vertical when aspirating the double deionized water. Therefore, any variation on the angle of holding the pipette will adversely affect the accuracy of the calibration. There are some brands of pipettes where the tip cone is held at a particular angle, and that angle can be maintained during the pipetting process because of the way the pipette has been designed. But in the main, most brands of pipettes do need to be held vertically when calibrating the pipette. The reason that you get inaccuracy when you introduce pipetting angles is the air cushion above the liquid in the pipette. When you aspirate liquid and you hold the pipette perfectly vertical, you will get a meniscus at the top of the liquid, and that has got a defined shape and a defined surface area. And when the pipette is held vertically, then that's how the calibration data is obtained. So you need to copy that technique in the lab when you're calibrating pipettes. When you hold a pipette at an angle, you increase the size of the meniscus on top of the liquid and the surface area. And unfortunately, that gives a greater surface area for the piston to act on when it draws up the liquid. So if you hold a pipette at an angle when aspirating liquid, you will tend to take up more liquid than you intend, and then of course dispense more liquid than you intend. So you will introduce a positive bias to the accuracy of the pipette if you pipette at an angle other than perfectly vertical. That's for most brands of pipettes.
Okay, fantastic. Thank you for that. That was great. I have another question here that is in relation to accuracy, and that is ISO microbiology standard deviations allow for plus or minus 2%. Is this too much?
It depends on the protocol that the lab is working to. Each laboratory decides what the limits are for the accuracy and the precision of the pipettes. It depends on what accreditation they're also working to. So long as the performance of the pipette performs within the specification that is in the operating protocol for that laboratory, then that's perfectly acceptable. Many labs work to ISO 8655. That's a good standard to choose to start with. So if the pipette is performing to the limits that are set in the protocol, then that is a perfectly acceptable standard.
Okay, fantastic. Thank you very much for that. Our next question here is, how could one maintain the pipettes to avoid error? Error-free throughput, the calibration time period, or error throughout the calibration time period?
Well, for most brands of pipettes, pipettes should be calibration checked at least annually. And then if the accuracy is found to be outside limits, then the calibration would be adjusted to bring it in within limits. How often a pipette is calibrated does depend on the accreditation that the lab is working to. And also, it does also depend on the liquids to some extent that are being pipetted. Liquids that that produce aggressive vapors, strong acids, alkalis, etc., they will affect the performance of the pipette, so that the pipette will need to be calibration checked and probably calibration adjusted and serviced more regularly. So in terms of a particular laboratory, then it would need to be established within the laboratory what time period would be appropriate for the accreditation that the lab is running, and also the samples that are being processed. I say most labs do seem to work to a 12-month schedule for calibration checking, but for many labs it should be more often than that. And I say it does depend on the liquids that are being pipetted in that lab, because some liquids do adversely affect the performance of the pipette quite quickly.
Absolutely. Okay, fantastic. Thank you for that. And this feeds into kind of pipette maintenance. And the question is, you mentioned two arguments for keeping kind of unused pipettes in a vertical position to avoid contamination, to keep them out of the way. But does this harm them technically if you keep them horizontally in a drawer, for example?
Yes, it's a little known fact that when you receive a pipette, you've purchased the pipette and it's arrived, the first thing you should do is check the calibration of the pipette to make sure that it's performing within specification. And then having done that, you should hang the pipette vertically on a pipette stand. You shouldn't leave it in the box on its side for any length of time, The reason being is that most brands of pipetters have lubricants in the inside of the pipette. They have O-rings which are lubricated. It's not all brands, but many have lubricants on the inside of the pipette. If the pipette is left on its side for an extended period of time, the lubricants tend to run down to the lowest point, leaving parts of the O-ring and parts of the piston, for example, dry. So that when that pipette is eventually taken out of the box and people start to use that pipette, you could well find that it doesn't perform within specification, and also the O-ring and other parts of the pipette could wear prematurely, affecting the performance. If the pipette is left on a stand vertically, then that effect is not observed, and it tends to remain accurate for longer.
Brilliant. Thank you for that. That's excellent. We have a lot of questions coming in, so thank you to our audience. It's great to see so many questions. We will try and answer as many as we can in this session, but any ones that we don't get to, we will follow up post-event, so please continue to submit your questions. Our next one here is, would it be possible to describe again the main differences between reverse pipetting and repetitive pipetting?
Yes, reverse pipetting is the technique where you take up the liquid that you want to dispense accurately plus excess liquid. So you take up more liquid than you need to dispense accurately, and that excess liquid remains in the tip. You are in fact working from a reservoir, and it's easier to dispense a viscous liquid accurately from a reservoir than it is to take up the liquid and then dispense all of that liquid out of the tip into the tube or microplate reproducibly. So the reverse pipetting technique is really a technique that means that you're dispensing from a reservoir. The repetitive technique is a form of joined up reverse pipetting, because in a repetitive technique, you take up the liquid that you want to dispense accurately, for example, master mix. You also take up slight excess. So you take up the liquid and the slight excess, and then you move the pipette to the first row in your microplate, and you dispense down to the first stop. When you dispense down to the first stop, you're dispensing the quantitative volume that you need to dispense accurately into the microplate, leaving the excess still in the tip. You then transfer the tip or the pipette over to the liquid that you're pipetting, and you then take up more liquid to be dispensed into the microplate. You take the piston from the first stop to the resting position. So once again, you have the volume you need to dispense plus the excess in the tip. And then you move to the second row in the microplate, and again, you just dispense down to the first stop, leaving the excess in the liquid. And you repeat that process until you've dispensed across the microplate. You will always have the same volume of excess liquid in the tip, and it means that you've dispensed accurately into the microplate using a technique that incorporates working from a reservoir. It is more accurate than using the forward pipetting technique, and it cures many problems. People dispensing master mix into a qPCR plate, they're using a multi-channel or sometimes a single channel pipette, and they're using the forward pipetting technique, and they're getting variation on the results that's down to purely the fact they're using the forward pipetting technique with a viscous liquid, with a sensitive technique. So using the repetitive technique should solve a number of problems in that scenario.
Brilliant. Thank you so much for that. And our next question here is application based, and it's which would be the best method to use when using whole blood in, for example, a blood film?
With whole blood, the most accurate way of dispensing whole blood, it's a difficult liquid to pipette accurately. It is viscous. It does contain red cells as well as plasma, serum. There are a number of techniques that you can use, and I think that in the laboratory you would establish which is the most accurate technique to use under your conditions. You can use the reverse pipetting technique because the liquid is viscous. So you take up the volume that you want to dispense plus slight excess, and then you dispense the volume accurately into the microplate or tube or the slide, leaving the excess in the tip, and that excess goes back into the original container. What's also a useful technique with whole blood is to use the forward pipetting technique as if it was an aqueous liquid. You take up the volume that you want to dispense, and then dispense that volume onto the slide or into the tube, and then repeatedly take up a small amount of the liquid again repeatedly, and then dispensing it back into the tube. So you end up scrubbing the red cells that were left on the inside of the tip and dispensing those into the sample. That's a useful technique as well with whole blood, which is quite unusual. So what I would do in that instance, I would try both of those techniques and see which one works most reproducibly under your conditions.
Excellent. Thank you very much for that. That's great. Our next question here is, do positive displacement pipettes have any of the accuracy and precision issues described for air displacement pipettes?
The errors that were described in the presentation earlier are errors which occur when using air displacement pipettes and the correct technique isn't used. A lot of the errors, pipetting viscous liquids, for example, the effect of temperature, the effect of pipetting angle, are really caused by the presence of the air cushion in the pipette, the air cushion between the sample and the piston. If the correct technique isn't used with an air displacement pipette, then errors creep in, and it's due to the effects that can happen with the air cushion. The air cushion can be stretched, it can be compressed, it can heat up, it can cool down, expand, contract, etc. That affects directly the volume that's dispensed during the process. With a positive displacement pipette, there is no air cushion. The piston comes into direct contact with the sample. So it's less prone to the errors that you see with an air displacement pipette when the incorrect technique is used. So very often it's the pipette to use for very difficult liquids or when pipetting in very difficult conditions.
Fantastic. Thank you very much for that. Our next question here is there any information or data available which will help me decide whether to use the forward or reverse pipetting technique for a particular type of liquid? How viscous does a sample or reagent need to be before deciding to use the reverse pipetting technique?
Now, this is a question that often crops up when you're talking about the forward pipetting technique and the reverse pipetting technique, because with many people, the reverse pipetting technique is completely new to them. It hasn't been part of their training in the past. The answer to that question is no. There is no data sheet that you can refer to. The decision to use either the forward or the reverse pipetting technique for a particular liquid must be made by the end user in the lab working under those specific conditions. If the accuracy and/or the precision obtained is found to be outside acceptable limits using one of these techniques, then the end user should use the alternative technique as part of the problem resolution. So let's just say that the end user is pipetting master mix, for example, into a qPCR plate. I bring this suggestion up quite often during this presentation because it is a scenario that people do find difficulty with because they're using the forward pipetting technique with a viscous liquid, and the reverse pipetting technique or the repetitive pipetting technique is the answer to the problem, and it does solve the problem of inaccuracy and imprecision quite quickly. Having established that the pipette is functioning well and the pipette tips used are a good choice for that pipette, the end user will find that the reverse pipetting technique performs best for most viscous liquids. So it's well worth trying the viscous liquid if it's completely new to you, if you're pipetting viscous liquids. And I am finding that in most labs or many labs that I visit, PCR is a technique that's used, and it's an area where people do have difficulty with precision and accuracy.
Brilliant. Thank you very much for that. Great response there. And our next question is, could you please give some helpful tips on how to select good tips for electronic multi-channel pipettes? I've come across a few situations where specific tips for our pipettes are out of stock. So do you have any tips here?
Pipette tip quality is all important. If you spend a lot of money on an electronic multi-channel pipette, then you shouldn't be scrimping on tips. So it's very important that the tips are of good quality and fit for purpose. We say that you should use pipette tips which are recommended by the pipette manufacturer for your pipette. Whatever your choice, you should try samples from available suppliers and do your own calibration checks or pipetting checks gravimetrically to test the accuracy of the combination of pipette and tip that you're using. Having established that, then you should have more confidence in terms of ordering those pipettes for routine use. So you should calibrate your pipette on the tips that you decide to purchase before you use them routinely. That should be part of the process as well. But calibrating the pipette with the tip should be part of the evaluation process. It's very important that the tip is a good fit, that it ejects correctly, and also it's the right polymer as well for the application. It may be that a low retention tip will deliver a more accurate volume as well, especially if you're working with viscous liquids. Also, if you're working with cells and dispensing cells using your pipette, then you don't want to damage the cells when you're pipetting. So you should evaluate tips which are designed for dispensing cells and have a wider orifice or wide orifice tips. So those should be thoroughly evaluated as well before you decide to purchase. Test the accuracy and the precision, and then you can buy with more confidence.
Brilliant. Thank you very much for that. We have time for a few more questions, so please continue to send those in. Our next one here is, where can I find a detailed instruction sheet for calibrating pipettes from Thermo? Is there more information available on this topic?
Yes, in fact, on our website, on the landing page for each pipette, you will find the user manual is one of the brochures which is listed, and you're able to view the user manual or download it and print it off. The user manuals are easily found on the Thermo Fisher website. If you have any difficulty or you can't connect through, if you get in touch with me or your local sales specialist with Thermo, then we'll be able to supply you the correct user manual for your pipette, certainly within a couple of days maximum. So that information is freely available.
Brilliant. Thank you very much for that. We have another question here, and that is, would you recommend a low retention pipette tip for dispensing 10 microliters of volume?
In terms of dispensing 10 microliters with a low retention tip, again, this is a question that does crop up quite regularly. In terms of Thermo Scientific manufactured tips and Thermo Scientific manufactured pipettes, we are one of the largest manufacturers of low retention tips. It's a big growing market, and we're seeing more and more demand. What we would recommend for your particular application, under your conditions, and for the liquid that you want to dispense, we recommend that you request samples to try, and we recommend that you do a gravimetric analysis of the volume that's being dispensed and thoroughly evaluate the tips before you decide to purchase. I do find that some end users have asked this question, and they have been given information that some brands of low retention tips are not suitable for dispensing 10 microliters and below accurately. But Thermo Fisher Scientific say that that's something that you would need to establish in your lab under your conditions with your pipette, and samples of the tips as well. And then once the suitability is established, then of course you'll be able to purchase them with confidence.
Brilliant. Thank you very much for that advice. Our next question here is, if you don't perform the blowout when forward pipetting, is the volume still precise? Aren't there leftovers in the tip?
If you want to avoid the blowout, you can avoid the blowout by keeping the tip above the meniscus when you're dispensing and still go through to the second stop on the pipette. So you are doing the blowout, but the bubble that's produced will not affect the sample because the bubble is produced above the meniscus. That is probably the most accurate way of dispensing. The purpose of the blowout is to blow out the last drop of liquid from the tip. What's worth trying is dispensing above the meniscus and going to the second stop for blowout. But also try dispensing to the first stop only using the forward pipetting technique and touching off carefully above the meniscus and seeing if you get equally accurate results under your conditions. So it's well worth trying both those techniques.
Fantastic. Thank you very much. We have another question here, and that is, what if pipetting of warm buffers or cold solutions is recommended in the procedure?
Well, if you know the temperature of the liquid that you're pipetting, then bearing in mind that most brands of pipettes, they're calibrated between 20 and 25 degrees centigrade. If you're dispensing liquids which are below that temperature or above that temperature, then you are likely to bring in an error in the process. You would need to incorporate the Z factor into the calculation with calibration to make sure that you're minimizing the error or bringing the error within acceptable limits. Ideally, you should equilibrate the liquid that you want to dispense to the same temperature of the pipette and the pipette tip. And I say for most brands, that's between 20 and 25 degrees centigrade. So the most accurate way of doing that is to just make sure that the liquid is within that temperature range. There are some brands that go down to 15 degrees centigrade, in which case then there wouldn't be an issue down to 15 degrees centigrade. But with most brands, and that includes the Thermo Scientific brand, then the temperature is between 20 and 25 degrees centigrade. And I would make sure that the liquid is between those temperatures for accurate dispensing.
Brilliant. Thank you very much for that. We have time for just one more question in today's session, and that is, can we be sent the slides for this presentation?
Yes, you can. We do supply copies of our presentations, and in fact, you will be able to view this presentation, I believe. So that wouldn't be a problem. And also, if you want us to do the presentation virtually at the moment for your laboratory or your company, don't hesitate to ask. That's a service that we provide. The pipetting techniques presentation is the most popular presentation that we do. It's absolutely ideal for new starters as part of their training. Don't hesitate to ask, and we will do a presentation for you that could be also tailored to your requirements. So yes, that wouldn't be a problem.
Okay, thank you ever so much, Steve. That's brilliant. Well, I'm afraid that's all we have time for today. So thank you again for your excellent presentation and your discussion there. And thank you to everyone joining us online and for sending in your questions. If you do have any other questions, please feel free to submit these now or email me at editor@selectscience.net, and I'll follow up on these with Steve. Remember, you can also download a certificate of attendance in the related resources tab indicated by the file icon on the left of your screen. And as Steve mentioned, if you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in a few days' time. So thank you once again, Steve. That was brilliant.
Okay, thanks for attending.
And goodbye, and thank you once again for joining us.